Started. Okay, good afternoon, dear participants. So, hope you had a wonderful break and good lunch. And hopefully the first two lectures in the morning was beneficial to you. Now we are going for the third lecture of the day, which is on TALEN. TALEN is transcription activator-like effector nucleases. And the speaker is Professor Ranjan Tamuli, who is my colleague in the Department of Biosciences and Bioengineering, as well as he is also an associate faculty member in the Center for the Environment, under which this particular course is being conducted. So, Professor Ranjan Tamuli had completed his MSc in molecular biology and biotechnology from Tezpur University. Then he went to CCMB Hyderabad, where he worked in the gene silencing in Neurospora crassa during his PhD years. If I remember correctly, under the guidance of Professor Kasbekar, and maybe Dr. Tamuli can correct me if I am wrong. Then thereupon he went to University of Florida for his postdoctoral research, and also he had another stint in the Florida International University, Miami, USA, as a postdoctoral fellow, and he joined IIT Guwahati in 2008 as an assistant professor in the Department of Biotechnology, which later on became the Department of Biosciences and Bioengineering. Professor Tamuli's research interest is on understanding the molecular process of calcium signaling using the model filamentous fungus Neurospora crassa. Professor Tamuli has won many awards and fellowships, and he was also awarded the Indo-US Research Fellowship by the Indo-US Science and Technology Forum for research at the University of California, Riverside. So he has many publications, and he has guided many PhD students and MTech as well as BTech students, as well as a couple of postdoctoral students. I have the privilege of having Dr. Tamuli as one of my colleagues and collaborators. Together we have done many projects, guided PhD students. Yeah, so with this small introduction, I welcome Dr. Tamuli to today's session and request him to deliver his lecture in this ATAL FDP on genome engineering for environment. Professor Tamuli, the stage is all yours.
Okay, thank you, Professor Bora, for a nice introduction. So good afternoon to all the participants. So today we'll be talking about TALEN. So although it is like online conference, but for me, like our regular say physical meeting, so I feel still say nervous, right? So even though it is online, so I hope I could share a few certain things with all of you, and I will get some opportunity to learn from you as well. So in today's research world, so all the labs except probably the people working with the computational field, they at least use certain kind of say some kind of restriction enzymes, so say nucleases. So without using say any restriction enzyme, it will be like very difficult to imagine by say biological lab. Now, if you just take an example of our human being as a model or as a system. So we all know, like, we have our length of the DNA is like very huge, right? So just a second. Okay, that's not. So our DNA is so long that you can think of about 70 trips from the Earth to the Sun and back. So that much of lengthy piece of DNA is accommodated in a tiny cellular environment in every cell in the human. And the situation is, and that is possible because the DNA that we call like our genomic DNA is very nicely organized in our so-called chromosome or chromosomal structure. So starting with like very primitive DNA double helix, which is around say 2 nanometer in thickness. So that will go on at some initial round of packaging. So we call them is like a beads on a string. And then this second level of this organization gives about say 11 nanometer. Then the very well-studied structure, next level of structure is 13 nanometer chromatin fiber. And then this will undergo the further rounds of packaging. It gives us like a 30 nanometer fiber. Then next level is 700. And finally we get the 1400 nanometer, the very popular, the mitotic chromosome. So we all familiar with DNA. So starting from like our school student, at least all of us have today, they know something, say DNA. And then chromosome is very familiar. So what we take home message from here, from these two slides, is that the length of the DNA is very long. And the DNA itself is very tightly packed inside our nucleus in the form of say, chromosome. So when this DNA undergoes several rounds of packaging, so now what is the purpose essentially fulfill is that it can store the information. So it can store the information in a very compact small environment. So if I have a suppose very long thread, if I roll around, so if I imagine this is like a long thread, I can roll around with a say ball, and I can use like a very small tiny space to store it. So you all have seen like electrical wires that are laid on this roadside. So when they are transported from one place to another, so you must have seen like those rolls of the wires, right? So once they are laid out, they become so long. So one purpose is the packaging is that it can store the genetic information in a small, tiny, compact environment. But the problem arises like a release. So because it is now very tightly packed, now because of that reason, so DNA, the information, whatever is stored in the DNA, so it will be very difficult to release the information on time. Because in order to release the information, so the specific DNA has to be accessible to the transcription factor. So transcription factor must be able to gain access to the promoter of a particular DNA so that it can activate the transcription of the target DNA. And that is crucial for say all kinds of biological activity, starting from fertilization till death of an organisms. And that information, a particular piece of DNA, also need to be accessed in a particular given time in a developmental time-bound manner. So that is one round of challenge is that from the transcription factor has tremendous difficulty in accessing the target DNA, and then it has become equally very or even more difficult for us to manipulate the specific DNA from using some say laboratory techniques. So first of all, so it is very easy for us to say imagine, for example, a nice circular plasmid. So you all familiar that we can clone our piece of DNA in a plasmid, and then it can be transformed into bacteria, say for example E. coli DS5 alpha, and then we can say express express it, or we can do it use it for some other purpose. But when you want to suppose specifically modify certain DNA inside the living cell, so that has become like a challenging. So how to give, say, specificity? So transformation and so DNA, introduction of DNA into a cell is not a problem. So there are a lot of techniques available. Say for example, electroporation, right? So we can easily insert or easily put a foreign piece of DNA into a cell. But how to give specificity for the integration, or how specifically a certain DNA should be mutated, or certain DNA should be corrected, or certain DNA should be or genes should be rather say activated, that has become very challenging. So therefore, to overcome those challenges, various types of techniques has been evolved over a period of time. So one of such technique is like TALEN that we are going to discuss today. So when one challenge, so we have discussed two challenges. One challenge was like for in a cell, so it is very difficult for the transcription factor to gain access to a specific gene. That is number one. And then in number two, it is very difficult for us to specifically modify the gene of our interest in vivo. And then third, another process. So both these 1 and 2, process 1 and 2 requires we have to have access to a particular DNA, and then there are also certain things goes on inside a cell. For example, DNA breakage. So when does DNA breakage takes place inside the cell? There are always roughly about say 20,000 different type of DNA breakage goes on in every human cell. And this type of breakages are always again repaired. So this repair must be takes place. If they are not repaired, then it will be deleterious for the cell, and then eventually it will lead to apoptosis. So to take care of this repair, the famous there are many process available. So we know the suppose homologous recombination, one such example. So apart from that, there is another some other group of polymerases are also available. So these are also called say translucent DNA polymerases. So these type of polymerases are also called error-prone polymerases. That is because so when the so you all know that for our regular type of DNA replication, the polymerase responsible for say for example eukaryotic DNA polymerase eukaryotic DNA replication is DNA polymerase delta. So now apart from that main polymerases, there are many other DNA polymerases. For example, DNA polymerase eta, DNA polymerase zeta, DNA polymerase kappa, iota, etc., etc. So these type of polymerases are called translucent DNA polymerases or error-prone DNA polymerases. So these polymerases are also responsible for repair of DNA breakage. And these type of polymerases are very important, or they play a very critical role in various repairing various type of DNA damage. For example, DNA damage caused by the UV or the ultraviolet, they are also equally important for cellular metabolisms, then also required for essential mutagenesis. For example, somatic hypermutation that is essential process during the maturation of our or during the diversity of our antibody diversity. So now, just now we have seen that DNA breakage, if normally, if there is a DNA breakage, so that is taken care by translucent DNA polymerases. Suppose there is quick DNA breakage, so it could be quickly repaired by a group of polymerases called translucent polymerases or error-prone polymerases. So we call them as like a translucent or error-prone because when they repair this type of DNA break, they also introduce or insert mutation there because these type of polymerases do not have proofreading activity, like the normal DNA polymerase delta. So in the absence of high fidelity or the proofreading activity, they tend to insert mutation, and therefore at the cost of repairing DNA breakage, they also introduce mutation, and therefore they are all very important for processes like somatic hypermutation. But when this breakage is severe, suppose both strand is broke down, double stranded breakage, then the cell has the option. There are two options. Either it could go for homologous recombination or HR. That involves the interaction between homologous sequences. So if there is, suppose both two strands broke down, we call them like a double stranded break or DSB, and if that happens, so cell has an option to go for homologous recombination. That is like a preferred one, because the homologous recombination is like a safe repair process. So it involves like interaction between the homologous sequences, and it is essentially error free. And this then if that is not possible, then there is another option available that is called a non-homologous enjoining, NHEJ, and this process involves direct ligation of the strand ends independent of DNA homology. In the like a simple eukaryotic system like Saccharomyces cerevisiae, so this they use an homologous system of the double stranded break repair homologous, and in which like exogenous DNA fragments are integrated at homologous site in the genome if the DNA has a short region of homology at both ends. So here there are many genes involved. So one of the very well characterized genes, RED52. So essentially, like homologous recombinations, in order to take place homologous recombination, the broken DNA in must be able to find out its counterpart, the homologous DNA sequences. And for like a simple organism, the Saccharomyces cerevisiae, where very few genes are there, right? About roughly around slightly more than 5,000. So whereas human, we have say about over 25,000. So where genome size is small and genome size is small, so this process can work efficiently. And in Saccharomyces cerevisiae, sequence homology of as short as suppose 20 base pairs sufficient for introduce homologous recombination. So therefore, so some of you must be working with Saccharomyces cerevisiae in your lab, or some at least work with Saccharomyces cerevisiae at some point of time. So when we work with Saccharomyces cerevisiae, we find that it is like very easy to manipulate the gene, right? Because we can design the primer of our choice, and then we can say go for transformation into the Saccharomyces cerevisiae competent cell, and we can just select the transformant on the basis of the selectable marker that we use. And it is very easy to modify the gene of interest. You can do like a lot of editing, a lot of modifications there. So because that is homologous system, recombination is very efficient there. But in most of the organisms that we handle in our lab, or the different labs or the people interested to work with, including human, plant, and insect, etc., this homologous recombination is not a preferred way of double stranded break repair. So this type of system, suppose the most organisms, the human, plant, insect, etc., so they preferentially use double stranded break repair. So this is because of several reasons. It could be, for example, the size of the DNA is also very big. So we all know that there is like a multiple origin of replication, and if during that there is some double stranded break, so cell need to quickly repair it as soon as possible. So you can't wait for searching for another homologous sequence. So if you go by like a probability, what is the probability that when there is a double stranded break, what is the probability that it will get another counterpart in say within a given frame of time? So that is very, very low. So so in normal situation with an organism with a very big genome size, it is always difficult to get the homologous sequence before apoptosis starts. So the so therefore the preferred way of this double stranded break repair is NHEZ or non-homologous enjoining process. So therefore, so this was the one of the reason why so initially the scientists or the people, different people, they started putting enormous amount of time and effort to find out some tools so that they can selectively manipulate or selectively edit or selectively knocked off or selectively suppose knock in. So there are so many different terminology. These are all confusing. Suppose sometimes some people would like to say overexpress the gene of interest, or sometimes people want to like say knock out. The knockout means completely delete the gene of interest. Knock in that is like some mutations, some kind of mutation. Or suppose sometimes they want to go for conditional expression of a gene, particularly if it is essential for a certain function. So there are many genes that are essential. So when we say essential gene, what does it mean? That the gene function is so crucial for the organism is that so if you directly knocked out that particular gene, it will cause like lethality. So if the organism will die. For example, in the mouse, there is a gene called say DNA methyltransferase 1 or DNMT1. So if when people knocked out that the particular gene in the mouse by creating transgenic mice, that mice was dead. Now eventually at in the embryonic stage. So suggesting that that particular methyltransferase is essential or vital for the cell function. So many such genes are so crucial that we can't simply knock it out and to study its function. And not only that, many times the organism also suffers the say foreign attack. For example, say viral disease, you know, the very familiar one. For example, say HIV, retroviral in human. So organisms, so therefore, the people are interested to modify or edit a particular gene, either because they would like to study a gene specifically, or they would like to say design some therapeutic intervention to prevent say foreign viral attack or pathogen likewise. So therefore, targeting a specific gene inside a cell has become like very crucial for either for the classical research or the basic research or application of on point of view. So that's where so that's how like so when we say for like editing, so when we suppose we want to edit a manuscript. So when you go for like editing a manuscript, scientific manuscript, we all are familiar with like publishing paper. So when we edit it, so as per the journal format, we have to keep in mind like the word limit, proper use of grammar, and say referencing style and such and such. So editors of a particular journal, they ensure the quality, and they will ensure like whether this particular data is going to fit for the journal or not. So likewise, so when when it comes to like editing our genome, so this editing also need to be very, very precise. So to make for that kind of precision, so the number one requirement is kind of say nucleases. So as the name stands for, so ASC. So these are like enzyme. So the enzyme, and these are enzyme type of restriction enzyme or the enzyme kind of group of enzyme or the molecular scissors. They are like a specialized protein that aids recognition and cleavage of specific DNA sequence. And so like discovery of the restriction enzyme was like one of the turning point in the field of molecular biology. So we all use them in a day-to-day laboratory experiment for many say variety of reasons. Starting from say most popular one is like cloning. So we are all must have cloned at least one gene in our life science research at some point of time. So we use at least the some kind of type 2 restriction enzyme at some point of time in our life. Right? So now and then the other two requirements is that like efficiency of homologous recombination is like increases the increases of the more than 5 orders of magnitude, so driving from 0.1% to 20% in mammalian cell with nucleus-based gene editing. So what exactly means that? So suppose if I take a cell, for example, we work in a neurospora crusher. It's like a eukaryotic system. So just now we have given example of Saccharomyces cerevisiae. So like Saccharomyces cerevisiae, so our model organism neurospora crusher is also a eukaryotic model system. So it is a fungal model system. So if I simply take some neurospora cell and do suppose electroporation. Electroporation is a technique through by using that technique we can insert the foreign DNA into neurospora cell. So if I simply take a suppose vector construct to modify or edit a particular gene in neurospora and do the electroporation, so percent chances of getting like homologous recombination is less than 10%, less than very less, very less. So although it looks like that means 90% of the cell transformant I will pick up, it will come as like unwanted type, and only 10% of the cell will be like a desired one, and so on. And although 10% looks in case of neurospora crusher, although 10% looks like a big in number as compared to mammalian cell, but still it is tedious because screening so many different hundreds of cells will require time, effort, and also the resources. So same way, if someone like try to do similar type of experiment, so in that case, mammalian cell, because the genome size is even more complex, it is like it's much bigger. It has more frequency of the introns. So every mammalian cell DNA, if you see like, although the protein is length is similar with respect to like primitive eukaryotic system, but genome at the genome level, the genome complexity is much complex as compared to other. So for example, we work with a gene called say neuronal calcium sensor 1 NCS. So if you take the protein sequence, what is NCS1 from a human and the rat and the say neurospora, so they will look exactly same in length. So they are same number of amino acids, right? Around 141 or something. I don't remember exactly, but it is length is equal. But if you go like a genome organization, like how many exons and introns are there, so there is like a huge difference. In neurospora crusher only presence of few introns, whereas in mammals and rat there are over say 50 different types of introns. So number of introns is very high in case of mammalian. So that's how the size is also very big. So therefore, in the efficiency of the homologous recombination or getting your gene into a desired location, also efficiency or probability also goes down. So therefore, when we use so when we want to improve the efficiency or increase the efficiency, when you will take a help of a particular group of nucleases, so the efficiency goes up to, so some say 20% in case of mammalian cell, if somebody is working with mammalian cell with nucleus-based gene editing. So that's how this has become very popular. And so it has given some kind of booster dose to the genome editing technologies. So in one line, so genome editing is like introducing desired changes into genome. Desired changes. So people could be like thinking of repairing some gene. Right? Say for example, those who are interested in gene therapy. So they want to like desired insulin, or they want to suppose prevent viral attack. Right? So they somebody who may be interested say HIV replication or HIV gene should not get expressed, or some other say viral disease, so on. Then the genome editing then harness programmable nucleases to cut and paste genetic information in a targeted living cells and organism. And so it is like a programmable nucleus. That means it is designed in such a way that where to cut and paste genome information. So in the first slide, we have seen that we have very long piece of genetic material, and thousands of genes are organized there. So among that, so many thousands of genes. So precisely finding our target gene and modifying only that one and not disturbing any other gene is like a key to our key. It will be the key. Suppose if you will have a technique that is doing precisely kind of changes in the gene that you want, but at the same time, if that also get inserted into some other places, right? So that will be a problem. So our essential goal in mind that there the changes or the mutation we want to do that should be specific only to the desired location, not anywhere else. Right? So it should not disturb or it should not cause any deletion or insertion at any other place other than the targeted gene. So that is the specificity of the technique is desirable. Now, so when we discussed that this type of situation, for example, the nucleus is given very say specific power to modify, edit and certain gene and a specific location. And people working with like similar line, they have also discovered the key component that is responsible for both two types of integration process. One is called say homologous, other one is non-homologous. So just now we have seen that when we have like a double stranded DNA breakage, so cell has an option to go for either a homologous or the non-homologous repair. So sometimes it is also called repair, right? So now when we take your DNA for doing any type of editing, any type of anything, suppose you take a foreign piece of DNA. So we make suppose nice gene targeting clone with all the desired properties there. So when we try to introduce in a cell, so we all so far discussed is that so it can either go to the directly the gene of interest, that is like a homologous integration, or it can go to anywhere in the genome. And so that will be called as like a non-homologous integration. So the key component, just now we sometime I told like the red 52 in the east. So this is the like a red 52 gene. So when we introduce a gene in the cell, in the eukaryotic cell, so we follows either the left hand side pathway or the right hand side pathway. And here in the boxes, these are the given genes that are essentially the east homolog, like red 52, red 51, red 54. So time being, we can just ignore the other one, the mass 51 we will just keep the simple thing so far. So essentially, so this homologous integration based integration primarily or the majorly follows either this pathway, red 54 dependent or the MIT dependent, or like a small fraction, in a small fraction of the cell, there is also independent of this protein. Independent of this protein also, the homologous integration can take place. Similarly, in the case of non-homologous integration, so there is something called say KU70 and the KU80. So Y is for the east. So if you focus only the last two letters, it is KU70 and the KU80. So this KU70 and the KU80 was were initially discovered in human patient. So this 70 and 80 stands for like a molecular weight of the protein, 70 kilodalton and 80 kilodalton respectively. And these two protein essentially bind to the broken DNA and terminate. So how this DNA breakage takes place? When you try to introduce foreign DNA, we go for, say for example, electroporation. or right, for example. So that causes the DNA breakage because we apply very high voltage. So that is the source of the DNA breakage. So once DNA breakage takes place, so broken DNA end must be immediately protected by the help of some kind of protein. So here is the rule of say Ku1780. So they bind to the broken DNA termini so that these broken DNA termini are not degraded by the nucleases present in the cell. So once this protection is given by this protein, then there is one ligase enzyme. So for DNA joining, so we at least know that when you clone our DNA in our lab, so we have used, for example, say T4 DNA ligase in our routine DNA cloning. So we all know that there are two types of DNA ligase. Either suppose E. coli, one is from E. coli, and the other one is say T4 DNA ligase. So these two ligases we are all familiar in our day-to-day routine cloning experiment. But there is also another type of ligase that is present in a cell. So in human, so this ligase is called say ligase 4 in human. So in human cell, so there is a ligase called ligase 4. So this ligase 4 is responsible for non-homologous enjoining. So when you, so in some people, some way back in say 2006, so what they have done is that essentially they have knocked out the human homolog of the ligase 4 in Neurospora acid. So that in so the homolog of this ligase 4 in Neurospora is mass 53. So when it was knocked out, so what one could expect is that because this ligase is not present anymore, so therefore in that particular cell, so this homologous integration pathway cannot take place. So this process. So in absence of this, so if there is no ligase 4, so there is obviously no homologous non-homologous integration. Only way the integration takes place is like a homologous integration. 100% of the time, the cell will go take repair the double stranded break only by the homologous integration. So that was good for like targeting, gene targeting. So in that particular background. So that was like a one of the big success in the like small organism, small say say lower eukaryotic system. But this is good for like a lower eukaryotic system. But again, for human cell, it will not be possible to say directly knock out the ligase 4 enzyme in the human cell and do our gene targeting. Because as I said, when we want to edit the human genome, so our editing we have to take care is that we have to take care. One thing is that we are not disturbing any other gene. We are just selectively mutated or selectively modifying, editing only the gene of our interest. And plus, the ligase 4 is also essential. Right? So if you suppose knocked it out, so without using nucleases, it will be easy for us to manipulate or edit the genes. But this is going to be like a deleterious. It will be harmful. So that is why we have to like so focus on other way of editing the gene. So now, so that's how. So this type of, as I said, the origin of gene editing comes from the homologous recombination. So people always are interested to homologous. So every time when you say the homologous is there because we want to the primitive construction is always done in the lab, right? Always done in the lab. So for example, we want to target edit any gene in the inside a cell. So our say preliminary work start in the laboratory. So we think about it, then we try to make some kind of construct using a suitable type of plasmid. So once our construct is ready, then we will only go for like inserting the plasmid or delivering the plasmid by using some kind of technique inside the cell. Right? So when we do our preliminary work in the lab for editing a gene, so in that particular plasmid, there will be a part of the DNA or the part of the DNA so that that plasmid so that that particular DNA can guide the plasmid to a precise location for integration. So that means in a plasmid, we must provide some kind of sequence homology as a guide, or to for targeting purpose, so that it can so that our construct can integrate a desired location. So therefore, it relies on the delivery into the cell of a DNA fragment carrying homologous sequences upstream and downstream to a target location, and the information it contains is inserted in the genome in the target location. And then limitation, of course, like inefficient in modifying genome, and then the solution has come like generating DNA based target locations. So what we so quickly can see. So in the right. So in our classical system, suppose we want to have a like this type of plasmid construct. Right? So we have suppose use a very traditionally, suppose in middle, this type suppose a plasmid. Right? So in the 5' side of the plasmid, we have suppose a blank box that we have seen. So this is in most of the cases, for example, the organism that we work. So we have here 5' flank of the target gene, and this right hand side is like a 3' flank plus gene, and middle of these two flank we put like a desired cassette there, whatever cassette should be interested there. So when we use this type of plasmid, both this 5' and the 3' flank act as a guide or the target this middle part to a desired location, and then it will get suppose undergo homologous recombination there, so it will provide sequence homology there. This will provide homologous sequence there, and then find it as a after this event. So we get desired cassette to be integrated in that particular gene. Now, so this so in this case, the success depends like how long, what should be the length of this flank. Right? How much length we should keep it, like a 5' and a 3'. So here, then the other problem is that if I have suppose 10 gene, then for all 10 genes I have to amplify the 5' flank and the 3' flank separately and keep cloning one by one. So that means it requires like a lot of cloning process, tedious, so many cloning, so many say ligation reaction, etc. Then so all this cloning again, when we say this these flanks provide the target, on the in another words, what we can say that this flanks is helping to generate the break, right? So it ensures that the break is happening at a particular location. So then the people thought that why not to suppose say introduce the break directly without using some kind of say nucleases. So that was like a generating DNA break at the target location. So without using suppose this 5' flank, if it is possible for us to somehow introduce the break at the desired location, then the life will be much easier. It will be more easier because we can eliminate the tedious cloning process there. And then without using this so many cloning steps, so one can do like introduce, or one can precisely control where this break should takes place. Right? So once the DNA breakage takes place at a particular location, then the rest part is easier, because the cell will try to repair it by using some means, and at that point of time, depending on the availability of the desired fragment with a particular sequence, so it will be easy for you to suppose introduce that desired DNA at that breakpoint. So it is like introducing generating DNA break at a desired location is like a very key for say editing a given gene. So now here again, so this part already we have checked, right? So when we target break by using some kind of nucleases, so cell will have like various options. So it could be like, so once break is done means so our job is 50% of the job is done. So we have done, say we could able to introduce break precisely at the position or the location where we want, right? So that is done. Then the cell will have two options to exercise. So either it can go for like a homology directed repair or the HDR. So in that case, the requirement should be the homologous DNA fragment should be available. So if that is not available at that point of time, so it can go for like a precise DNA modification is possible. So on the other hand, in absence of that, if it follows, like suppose non-homologous end-joining pathway, and then it can lead to like a variable DNA insertion or the deletion, gene inactivation. Right? So inactivation. So those who were like at least design some say expression cassette. So we all know that when we design any gene for our say bacterial expression or eukaryotic expression, we always also careful about suppose even a single base, right? Because inserting just one single extra base or missing one base in front of triplet will lead to like a in-frame stop codon. So that will like a so that will cause like inactivation of gene. So same way, the non-homologous end joining would eventually result in like a variable DNA insertion or the deletions, that is also called like gene inactivation. So therefore, like double-stranded break are introduced by nucleases engineered to target a specific DNA locus. So in all kinds of our list, TALEN and other things, so we basically engineer, these are like engineered nucleases so that they can introduce the double-stranded break at the position that we are interested in. So all this, then thereafter, the life goes on like simple. Right? Either it will go for like homologous based HDR or NHD pathway, and depending on the which pathway it follows, we get output. Either we get like a precise modified DNA or like a mutated version of the DNA. So this homology directed based repair, repair that we know. Right? So inside genome break leading to precise DNA modification. The other one is like in other case, it will be like error kind of error prone. So in the beginning of the slide also we have seen that when suppose there is a single strand of DNA is broken, then in that case, if you have only one strand is broken, then it is translucent DNA polymerases, right? So or the error prone DNA polymerases. So to repair those kind of single strand break, so we have like polymerases in the cell. We call them error prone polymerase or the translucent DNA polymerases, and they repair at the cost of inserting mutation. So similarly, this non-homologous end joining methodology, when there is both strands are broken down, yeah, so if the cell uses this non-homologous end joining, so this in this case also the resulting output is or resulting DNA error will contain some kind of mutations, and therefore it is like error kind of error prone mechanism. So resulting most of the cases will inactivate the gene and gene, and it can lead to like a frame shift mutation or a premature stop codon isolate if there is a problem even like a missing a single base or having like one extra will cause like a in-frame stop codon or a premature stop codon and leading to potentially dysfunctional repair. If you have a quick look in our this timing or the chronology anything. So like a long time back, like we all know like each HO endonuclease that was like a very famous for yeast mating type switching, and like yeast we all know that this A and alpha mating type, so it can switch the mating type, and there where like HO endonuclease play a very key role. And then after long gap, 1996, the testing of ZFN. So we have a talk in this QIP. Then gene correction in human cells via ZFN, zinc finger nucleases. Then targeting genome rearrangement using ZFN. Then again, the other one, very popular and the famous ones are like in vitro reconstitution of CRISPR-Cas9. So again, this is covered in this QIP. Then gene regulation via CRISPR-I and CRISPR-A, then genome editing with like CF1, then boosting homologous recombinant NHZ, then Drosophila genome editing 2012 via ZFN, and then 2009 cracking of tail code, this is in 2009, then genome editing with human cell using TALEN, then human genome editing using Cas9, then RNA guided epigenome editing, so the latest one. So TALE is like a relatively new. It is a relatively new technique or the relatively new mechanism to edit the human or the let's say mammalian genome or the eukaryotic genome, rather to say, and then like other techniques or editing. So this also basically serve the main purpose is to efficiently edit the gene of our gene at a precise location. So all these methods listed here are by and large same, except the last one. This is like a RNA guided epigenome editing. So here it's like a epigenetic editing. So we all know that mammalians type of DNA is like even more complex, because in the case of mammalian, or say including our human, so DNA, so all cell functions are not simply governed by only the DNA sequence. So many of the times the epigenetic status of a particular gene. So for example, say methylation status, the very well known is suppose in cancer, or say suppose a suppression of cancer. So there are many genes called say tumor suppressor gene. So at normal time, those suppressor genes are active, and when there is like a say transforming cell or cancerous cell, so those tumor suppressor gene get silenced by this methylation of their promoter. So in those cases, if somebody would like to suppose edit the gene, that particular gene using TALEN or some other technique, so it will not yield any result. Right? So in that case, one has to basically try for say changing the epigenetic status. For example, remove those methylation marks so that the tumor suppressor gene can again get expressed. So in those cases, this RNA guided epigenome could be like a very promising in coming days and so on. So every technique has like their own ways of action or the mechanism, and they have like a different way, but essentially all are like harnessing the molecular biology tools or the knowledge for better understanding of the genome for therapeutic purposes and so on. So here, as you said that these techniques are by and large basic purpose is same, like all these nucleases, mega nucleases, zinc finger nucleases, TALEN, CRISPR Cas9. So all this system, the goal is to like somehow introduce double standard break. In one of the slide we have mentioned is that the single standard break could be easily made at any position, non-specifically. For example, popular one is suppose UV. So UV can cause single standard DNA break, right? And break. But now to precisely introduce double standard break, there are so many tools already available. So one can nicely like choose any one of them for their work or the application. And once that is done, so cell will go follow any of this methodology or pathway for the repairing double standard break. So if it goes for, as we discussed like in previous slide, we have seen that if it is like non-homologous type of or the pathway of the repair, so this will cause this may cause like insertion and the deletion or like a disruption of the gene. But at the same time, when such a double standard break is there, then at the same time somewhere, and then suppose one template is also available, some kind of targeting template is available quickly. So there is a good chance that this available template will get quickly like it there, right? Like it there. So so in normal situation, and so before discovering of this type of technique, what people used to do is that people simply also make a vector construct like this. So that's now we have discussed, right? Vector construct. So they will provide suppose 5' flank of this particular gene. Suppose this is 5' flank. Suppose this is the 3' flank of the target gene, and they want to introduce some kind of foreign DNA and there. So they used to simply make this type of plasmid construct and do a trial and error. Suppose they simply do a transformation and then see whether the homologous recombination is occurring or not. And because in case of the large genome, say in mammalian cases, the chances is zero, probability is less than 0.001 and etc. Right? So this tool, but this tool, for example, this tool at TALEN, this nucleases, because this nucleases is like an engineered version. So because they can introduce a double standard break at a precise location. So therefore, the chances of getting the desired or getting or the editing the desired gene, there has become the potential increases tremendously. So in the previous example, so in this example, when we use a classical type of plasmid, we said that, for example, we use electroporation. So when you do the electroporation, high operation, then we have no control, like or no control of the DNA break. So in that type of technique can cause the DNA break anywhere, right? So it can cause DNA break in our desired location as well as multiple other places, So places, and all repair is going on. So when there is a multiple suppose when there is suppose double strand break at multiple places, then there is a very high risk of having this one, say non-homologous enzyme, because we are providing only one template normally at one point of time. So so in the classical type, therefore there is number one, the probability of the chances of getting precise insertion is also low. At the same time, having this type of error or the mutation is also very high. So now, because we have specifically engineered nucleases which can introduce the DNA double break only at the site where we want, not anywhere else, not here and there unnecessarily. So therefore, because of that reason, so we have like a less chance of mutations. Rather, we can, however, very safely or very precisely control the editing only at the target site, not at some other position or the location. So this how like this gain huge attention, and so many people got interested in this technique in recent time. So this transcription activator-like effector nuclease or TALEN. This is again like editing tool, and it's a good alternative to zinc finger nuclease and the meganucleases, because even they can go so specific that it can choose like a single nucleotide. Right? So you can give very specific targeting. Then it comprises like a non-specific DNA cleaving nuclease fuse. So as I said, it is like an engineered kind of nucleases. So it has like obviously the last part is like nucleus. So this nucleus is like a DNA cleaving, then that is DNA cleaving nucleus, and fused to like a DNA binding domain. And this is like a two parts of the protein coming from two different sources, and that can be easily engineered so that TALEN can target explanation, which is nicely summarized here. So one is like a DNA binding domain, it's still, and we fuse with someone cleavage domain or say nucleus FOK1, and we get the resulting fusion protein is TALEN, fusion protein of fusion enzyme or the nucleus, which is TALEN. So so that's how we get it. So this is like something, say if somebody work with say is2 hybrid system. So in the experiment using like I say is2 hybrid system, we have like a specific DNA binding domain, and then we suppose fuse with like transcription activator domain. So when DNA specific when DNA binding domain binds the target location, so it can drive the expression of the target gene. So then we can see like whether this our construct is able to activate the transcription of a particular gene or not, etc. We can use a reporter. So likewise here, so we had like I said like a fusion or engineered protein or say enzyme or nucleus. So that is coming out like a fusion of two different. One is DNA binding domain, and another one is like a nucleus part, and together. So DNA binding domain is giving specificity to govern it, essentially where the nucleus should go. Right? So DNA binding is giving the specificity for the DNA binding. So you design. So when you design, suppose the TALEN for our application. So we basically need to design our DNA binding part. Right? So which DNA it should bind. The other part, what is the nucleus, is the common. So this part is so that is available. And then the resulting hybrid or the recombinant will give rise to a specific TALEN for a specific gene. So now 4 different repeat domains can be say mix and match to create new DNA binding proteins, which can be linked to the fork1 domain to create a new class of programmable nucleases. So we will see in details. So again, so now the tail is also, so here the first three letter is TL, tail is a transcription activator-like, and so it's like a activate. So from here, this word, what we understand, this is some kind of protein, so which can like activate a transcription of target DNA, and this is like a class of specific DNA binding domain. So because they are they activate transcription, so therefore without reading anything, one can imagine that they will bind to DNA, right? DNA means more specifically promoter of a target DNA. So transcription starts mainly say transcription control elements are mostly present in the promoter. So one can imagine that just before the ATG start codon, this tail transcription activator will bind. So there are many different transcription activator, and if those who are probably someone from non-biology background, so in eukaryotic genome, there are presence of so many transcription factors. They are essential. If you just go back to the like because of the reason that previous slide, that number one, say first of all, the DNA is heavily packed. So when somebody will try to unpack it, right? So it has to apply tremendous say energy and the force, and so because DNA is like a, you have to make a unwinding your DNA, and then to open it up the DNA, this activator will provide like a sufficient, it will open in such a way that the transcription factors, the main the polymerases, they can activate it in a say required threshold. So these are like a class of specific DNA protein, and these are basically originally found in the plant, the very famous bacteria, the Xanthomonas. So it causes a lot of disease in different types of plants, and we also see in the next couple of slides like RVD. It is say repeat variable dye residue. So these are very important residues, mainly present at 12 and 13 position, and this whole process involves either say a non-homologous enjoining or the homologous recombination that we have discussed already in the previous slides. So now this is like one pictorial representation so of the TALEN that we have present. So we have like in engineered cases, one N-terminal domain, and then tail repeat domains are there. So you can see like different amino acid different repeats. So here whatever we are seeing is that these are all different color coding, different repeats. Suppose 1, 2, 3, 4. There are so many repeats are there in this particular domain. And then one is a C-terminal domain, and this is the nucleases. This is the nucleases. So all these repeat in a simple sense. So these repeats are providing where it should bind. So basically the repeat determines the sequence determines the target binding site. So there are two such protein will bind. Essentially it is like a dimerization, and then the nucleases is the main part is the nucleases. Another important part, nucleases that will take care of the say creating double strand break or the cut. So here is here if you take one only, so I told you like so many repeats, right? The 1, 2, 3, so many. If you take just one single repeat from the tail repeat domains, so the sequences would be like this, something like that. So this is like a repeat. So in this example, and this is like HD. So if you count like 1, 2, 3, 4, likewise, so essentially 12, 13 this amino acid. So these amino acids are crucial for generating the specificity or the base selection. So you can, so people can design as these specific pairs to target the TALEN to the specific location in the genome. So in a simpler sense, so what is we understood here that it is like a fusion protein. This contains one say tail repeats. So this tail repeats determines the DNA binding, where it should go and bind. So the binding is determined by the sequence that is present in the tail repeats, and then the common part is HOC1 nucleus. So the nucleus, so once this nucleus, once this TALEN TALENs recognizes particular DNA sequences, then final DNA scissoring or the double standard break will be performed by the HOC1 nucleases. So it can do that double standard break. So as I said, this is originally derived from this Xanthomonas. So it is like a gram-negative bacterial plant pathogen. So it's a very smart bacteria. So this bacteria has various type of protein secretion system, typically naming like a type 1 and type 6 type of secretion system. And this is also like a pathogenicity is mediated by the hypersensitive response and the pathogenicity HSRP, or in briefly, it is called HSRP type 3 secretion, or TTSS. T for this type, T for like a type 3, and SS secretory system, right? Type 3 secretion system. So this bacteria is caused like a serious diseases in plant, right? Various type of plant. So it can, like you can see here, we have some representation. So there were, and there's a famous group working with Xanthomonas at CCMB. So I have also opportunity to see that. So when I was doing PhD there, so in that the Xanthomonas, so essentially causes so that group was working on the rice. So rice disease caused by the Xanthomonas in rice. So when is basically so here in this pictures, so bees, for example, like wheat infected with the Xanthomonas, and this is bacterial blight of the rice, and this is bacterial leaf stick caused by the Xanthomonas oryzae. So this is like Xanthomonas oryzae. Then this is for the rice pathogen. Then you have the wheat is Xanthomonas campestis, etc., etc. So all these species, they are basically for plant pathogen. So they infect the plant leaf, and they can successfully colonize this they can suppress the host defense system, and they can successfully make the host pathogen this barrier, and then finally cause the severe disease, and which can drastically like reduce the drastically cause the reduce the yield or eventually total loss. So if you compare like say genome size of the Xanthomonas versus its host, the host genome is like a more complex. So we took example our human, human say what we have heard in the first 2C slide is that they are very say big in size in layman word, and also this higher eukaryotic gene is also packed with various histone proteins, so that they can be accommodated in a tiny cellular environment. But this bacteria genome is like a relatively small, and then they are not also so packed. Right? So the bacteria have only one single chromosome, so they do not also have like any histone. There is no necessity for it to being packed in a tiny molecules. So what this bacteria do essentially, so they have like a very well evolved a different type of secretory system. So for example, in here we have like type 1 secretion system, then type 2, type 3, type 4, type 5, type 6. There are so many different types of secretion system in the bacteria. So they are responsible for different type of function. For example, this type 1 is required for this adhesion to the host cell. So the first requirement for any organism to being as a pathogen is like an attachment. Right? Attachment to a particular surface. All right? If the bacteria or any pathogen, if it cannot attach or recognize a host surface, then it cannot cause the disease. Right? It will simply will not able to make any interaction with its host. Right? Then, so first requirement is so this type of some proteins are required for this making adhesion to the host cell. So those proteins will be released by this type 1 secretion system. So similarly, there are other types of proteins are required for host cell was there. So once attachment is done, so all the cell has like a cellular membrane. So like plasma membrane in the mammalian cell, or suppose cell one in case of the plant. So in that, so those type of protein that are required for degrading those cell walls, so that once the cell wall is broken, then it can attack that particular cell. The pathogen can attack it. So there is a number 2 step. So it is like type 2 system. The most important one is like a type 3. This is like a host cell manipulation and host cell manipulation pathogenesis. So even if a bacteria is successful to get success, or pathogen is able to successfully attach to a particular host, or suppose it is successfully able to degrade a cell wall, but if it cannot manipulate the host, then it's a very good chance the host can eliminate the pathogen. So same is true for example the viral diseases, right? So when we suffer from, we know so many different type of viral diseases, right? So when researchers or scientists design any molecule or any drug designed to overcome the fight against those viral diseases, so we all are very familiar that over a certain period of time, the virus will basically derive or develop some kind of mechanism so that it can escape from that particular drug, or it can resist it. So same is true for bacteria as well. So this is like we all know about like bacteria, fungi, and so many. So we all know that the drug resistant bacteria, drug resistant fungi, etc., etc. We also know about the multi-drug resistance organism as well, MDR. So now, likewise, this plant pathogen as well also has a very good secretion system, so they can evade the immune system, plant immune system. And how would they essentially evade or escape is that by manipulating the gene. So if the plant is trying to suppress the bacteria by its defense system, so it essentially modify the host genome in such a way that it can become incapable of resisting the bacterial attack. So essentially the bacteria precisely know like which gene to basically mutate, which genes to get silenced. So these type of proteins or enzymes are secreted through this type of system. Then we have like other secretion system as well. So some of them are like type 4 involving bacterial competition conjugation. Then type 5 is required for addition to the host cell, also required for biofilm. And then also there's type 6 that is required for like a microbial competition, etc. So what we are talking about here in our TALEN cases, it is we are basically harnessing the DNA recognition from here, something we are taking from the type 3 secretion system. So this is like more detail of the previous slide. We are showing the same thing, like all these 6 type of secretory system for the different types of the secretory system, type 1, type 2, type 3, type 4, and type 6, and so on. Now, in original time when it was first discovered, so what it was understood is that this in bacteria, the transcriptional activator of plant genes. So in this case, in the case of the plant bacterial interaction, that means suppose Xanthomonas and the plant case, that example. So the bacteria first need to get adhere or get to the host plant leaf. So attachment has to be there. And then the first is like racing to the plant cell, and secretion and the translocation of effector protein in the plant cytoplasm through the type 3 secretion system and translocate complex 1 of Xanthomonas. So something is being secreted by this type 3 secretory system. So we say this is TAL effector, so which was initially inside the bacteria for the particular secretory system that is being now released into the plant cell. So now effector is inside the plant cell, and then finally this effector would able to localize to the nucleus of the cell. So because localization now the TAL binds to DNA and RNA DNA and then binds to DNA. So this is able to enter the nucleus. So now because these are secreted protein, so therefore they also need to have like a secretory signal should be there. So therefore, because they has to go to the nucleus, therefore obviously the nuclear localization signal or NLS should be there. It is not mentioned written here, but they should have like because the secretory protein must have like a secretory signal, as well as all nuclear protein, you will find like they will have like nuclear localization signal, so that they can become nuclear localized. And the job was like to, as I said, the main job or the function was to suppress the plant defense. So once they go and bind to a particular say DNA target sequence, so basically they were they could able to suppress the plant defense. So basically they will express something or they will do something there inside the nucleus so that plant can become defenseless. Right? Then also the modulation of the transcriptome to support the vector. So they are not only just suppress the plant defense, but at the same time they will also manipulate the this expression of the or the transcription of the certain genes, so that they are useful for the bacterial growth, the bacterial virulence. So it is essentially hijack the plant transcriptomic machinery for the benefit of the bacteria. So now this tail repeats. So tail repeats the longest effector family out of total 2040 type 3 effectors. This is just to give one example. The first evidence is isolated and Xanthomonas, and then we already have seen in our previous some slides, like in case of tail, the central repeat domain contains a lot of different repeats. So we have already seen that in the DNA recognition part, there are various type of repeats are there, and these repeats are responsible for providing the specificity, DNA binding specificity, and then also in the unengineered version of the tail will have your acidic activation domain, nuclear localization signal, in that it is localized to the C terminal, and this tail has one nice thing to look into characteristic feature is that this type of protein can recognize a single base. So because they can localize a single base, the advantage is that there is a huge advantage for the bacteria. The huge advantage is that because they can recognize a single base, so therefore it is very difficult for plant to fight against it. And even if the bacteria plant suppose is successful to do something against the bacteria, so bacteria will quickly mutate it. Mutate in such a way it can recognize another base, some other base specifically. So it is helpful for the bacteria to evade the plant defense, but is also now good for the researchers, because they can very nicely target it to a DNA just by using a single nucleotide. This type of slide we have seen already previously, like there are like multiple repeat regions are there. So in each repeat we have we give a repeat, and then we give the residues that are located in the position, suppose in this position, say 12, 13. So whatever amino acid is, you place it there. So this is like a single letter code of the amino acid. So whatever amino acid is present in there, so that pair basically determines like which nucleotide sequence it's going to recognize. So therefore it is called repeat variable diresidues. So this is like a repeat variable diresidues, or RVD. So this is RVD, repeat variable diresidues, and you can design like so many different types of these. So some examples are that if it is like a pair of N and Z, so it can recognize T. If it is a pair of N and I, as far as in an isoleucine, it is like a adenine. So this side is amino acid sequence, 1 or 2 also amino acid sequence, and this side is specific DNA, right? And this is histidine, glutamic acid, and cytosine residue. If it is NS, like it is, it could be A, Z, C, T, etc., etc. So there's this way by based on the specific nucleotide where we want to insert our cleavage, where we want our TALEN to be bind. So based on that, we can design this particular repeat or the specific things, and then it will be so we can essentially precisely control like which sequence or where does it go and on the recognition sequence, right? So this is same like a previous one. So this two sorted in the secondary, so it has level like alpha helical structures and so on in normal tail proteins. And here is the other one important part. this FOC1 restriction enzyme. So we have discussed that. So our TALEN or the recombinant nucleases or the modified nucleases contain two parts. One is like a tail protein, which is required for the DNA, specific DNA binding. And then other one is like a non-specific, say another one is like a general restriction enzyme or the type 2 restriction enzyme. So it was originally again derived from another type of bacteria, Bacterium. Okay, and this recognizes this sequence like GGATG. So we, as I said, we all use restriction enzyme at some point in our life. So this is also a similar type of restriction enzyme, but this cut about in the 5' strand, about 9 base pair away on the 5' strand and 13 base pair away on the 3' strand. So this is the sequence where you will get eventually DNA cut or the double stranded break. So this is the enzyme. So this can create the responsible for the introducing the double stranded break, and the other protein the tail, whatever we discussed so far, that is responsible for giving the binding or the targeting. So essentially, suppose if you simply take the FOC1 and if you express it, so it can create, say, a break at maybe at the wrong place, or we will not able to control it. But because we fuse this one, we have a specific DNA binding domain. So therefore, our engineered protein is able to like introduce the DNA breakage at the precise location, and this is all like a double stranded break. So this is again like a same thing. So this is shown like a middle, the 1213 HD residue. This is the residue responsible for this TALEN to recognize the sequence. So these we can just skip right now. So likewise, when this we design this TALEN protein, so real life situation, we know that the dimerization, so two repeats or two is like hybrid recombinant protein will bind, and when there will say hybridization, dimerization of this protein, dimerization of this nucleases, so they will start cleaving the both strand of the DNA, and then we get double stranded break at this particular site. So this two domain has given your where the DNA, the binding, recognition, specificity, and these two nucleases is responsible. These two nucleases is responsible for introducing cleavage, cleavage at the site of cleavage at the defined site. So that's how we get our desired DNA double stranded break at the location that we choose or we are interested in. Like for one. So in real life situation, so they work likewise. So same. So we have like we all can also call this like a left TALEN and the right TALEN, and then the functional domain is like your this nucleases. So they are responsible for introducing different domains, and you can also modify their same concept to do some other work as well. So for example, in this below one, so here suppose we are not interested to do like double stranded break, rather in this case, so here it is something else, say VP64. So this is like a transcription activator. So sometime back I told like give an example of say is2 hybrid system. So similarly, if somebody is interested to say harness the DNA binding, specific DNA binding properties of this TALEN to activate suppose gene of interest, so they can modify this same concept, and then they can fuse it like transcription activator, and in that case it will drive the expression of a target gene. So we can basically either introduce double stranded break for integrating with a gene of interest, or it can also basically can be utilized to activate transcription. So the advantage is that this type of cassette can be used to target any sequence in any cell, at least in principle. It can be any cell. Of course, there are limitations. There are data also depends on like where the cell is located, etc. But at least in theoretically, it is possible to target any sequence in any cell, and highly sequence specific genome editing. That is for sure. Like we all agree with that. That's like it is a very specific Then it is very good for like gene knockout, knock-in, mutagenesis, activation, repression, and more, etc. Right? So knockout is just we have to know that we have completely delete the gene. Knock-in, suppose you want to conditionally express and shut down the gene of expression. Mutagenesis, for example, we want to suppose change the 1 or 2 amino acid residues, key amino acid residues, to know like the functional significance of a particular amino acid. Or activation, this is the example like how to activate a gene of particular gene. Similarly, instead of transcription activator, if somebody would like to suppose they put like a transcription repressor domain, in that case, instead of a transcriptional activator, the transcriptional repressor will occupy this position, and then in that case, the expression of gene will be shut down. So it is shut down. So like transcription activator, the same technique can also be utilized to transcriptionally suppress or repress the gene of interest. Then flexible tail effector design of the binding and functional domain, such as, so this is like a TALEN TF. TF is a below example transcription. Okay, so that means this concept can be utilized for a number of purposes, in particular in the mammalian cell or the mammalian cell where the genome is more complex, genome size is more much more bigger than like a primitive or the simple lower eukaryotic system, and then you can have a like a very good chance of manipulating or the editing only the gene that you are interested in, without like altering or modifying any editing any other gene unwantedly. So in a, like in this cartoon, so we have now this is same like other type of common routine molecular biology approach. So we have to first like say think about it. We have to just first think which gene we are going to knock it out, I don't know, and design it, the experiment. We take the like various type of softwares, so software to design the construct, and then obviously the PCR. So we do the PCR, then we clone it, the routine cloning, TALEN gene cloning, then transfection by electroporation. I told like electroporation is like a very good technique to introduce like a foreign DNA in a say mammalian cell or the even fungal cell as well. If there is also other techniques available, for example, say transfection, lipofectamine-based transfection, you can use also that particular technique to introduce foreign gene. Then all this kind of thing like sequence recognition, then binding will take place. So we get either say double stranded break once, suppose YFG text, suppose you want to put it there. So double stranded break once it is done. So it will once it is done. So it can either go for like homologous based pathway or no homologous or the non-homologous pathway, and depending on which of the pathway they follow, it could be like a it could either end up in having some error or the mutation, or it could be like inserting some tag in the gene. Suppose we want to put a tag, or we want to put suppose label the DNA with some reporter marker, etc., etc. So your favorite gene, so YFG. It could be inserted likewise. So now there are, so these guidelines are not say that this is the only thing it is followed. There are various modifications. So in many papers, the repeat length is like given in a different numbers. So as a whole, like we have like a take home message is that there are multiple repeats should be there. And TALEN should comprise various type of repeat. So people can have say 11.5 and 70.5, so for example, and then nucleotide proceeding the regions region identity specified by the RBDs should be a T. Right? So just before the RBD, we know on the repeat variable di residues. So just before that, there should be like a T residues, and these are other guidelines like use. There are several other options. So you can use either say is the N and NZ NAC this amino acids for binding to the C, Z, T, A respectively. That means we have to first decide like where our cassette should go and bind. So where we want to introduce the break. So what is so based on that? Once we know like where we want to cleave it. So based on that, we can design our experiment. We can then based on that we can design our repeat combination of the dye residues RVD, and then by once this decision is made, so then that next the TALEN should be contain at least 3 strong RVDs HD order, and then avoid long series ANT in the beginning of the question. And so this way we can like a basic guideline. So you can have also like other parameters to be considered. So now that situation has become little more easier because we have access to various computational tool. So in a time wise, normally, although it is so simple, but in real life it may take some time more. But in general, so if you consider for a simple experiment, so at least one day could be like say target design one target. If we have a suppose or we are just ignoring the lower part, suppose we want to design decide one target. So in day one we decided one particular target and designing. So then your PCR and other things are done, right? So then suppose we get a primer. Of course in here we cannot get primer and our design cases so fast, but once you get it, so we can do all this cloning and etc., then DNA preparation, and then say pig culture, etc., DNA preparation, then we are doing transformation to get up interest. So as I said, now the life is a little bit easier because of this available of the various software tools that can be tremendously utilized or tremendously helpful for choosing, because this manually counting the base. Suppose if I want to suppose design one primer, so a primer, simple primer for the PCR primer for a mammalian gene. So although the manually we can design the PCR primer, taking care, suppose for example when we design routine DNA binding primer, so we suppose follow some guideline, say they should be suppose about roughly 18 to 21 nucleotide in length. Then suppose their end should be ending should be with a purine, suppose either Z or C, and we should not keep suppose same residues in suppose running into multiple. For example, in this software, suppose if you are looking at here about this place, there are multiple keys are going on. So when we design something in our real life manually, so it will be very hard for us to suppose if I see suppose there are so many keys here, then I will move to the next place and try to find another good sequence there. So it is essentially, it is possible, but we by doing to design primer manually, but it will be also laborious and time consuming. So we can do probably one in just one day. Yeah. But these computational tools are useful like when you are handling with a complex genome, particularly with the mammalian version, because the mammalian gene, as we know, that number of introns are very high, you know, so many introns. So manually we can do it for like a lower eukaryotics, but this computational will help you to deal with like complex genome like those one in the mammalian. So there is many more such tools. So one is like for a basic layman, maybe even I am also layman. So this talentdesign.org. So this is like a good one. So I find it's like a little bit simpler. So here you can go to this particular address. So if you in your free time, if you simply click into this this link, so you get a very nice say interface like this. So you can do a whole lot of things. So you can like select the genome against which you can design like short flank length, long flank length, right? So what is the flanking side? So you can actually have this type of parameters, and then here also you can choose like enzyme provider. So one restriction enzyme you have to buy it purchase, right? So from which company you are buying it. So although the restriction enzyme is in principle same, but in the you can but still their efficiency with a particular combination of the buffer system is could be different. So you in this web page you can also take care of the enzyme basically from where you are going to get it. So this is like a simpler one, and it's easy to use for at least for beginning and learning purposes. So like this talent design, there are so many others as well. So each has like a specific. So there was a nice paper. I think I have given the link somewhere. So there are, for example, this is one tool, TALEN, composer. So each has specific purposes. For example, here in this case, you can go by gene ID, search, fly-based for Drosophila, worm-based, Saccharomyces cerevisiae. So those are so this is specifically good for the people working in say Drosophila genome. They were also good for the people working in Saccharomyces cerevisiae. So then here this is designed for to work with those these genomes, and then like the other one, TALEN. So here you can go for also the similar you can go for TALEN, and then there is also the other one, Progno, Septa, TALEN, NT, then probably there is some more. I think, yeah, there is some more. Then again, TALEN designer. This is just now we have seen, like this is human, mouse, rat, genomes, etc. And the TALEN of her. So this these databases or the tools are like people have published in a different and they are optimized for a specific kind of work. So depending on our purpose or depending on the model system, so it is possible for us to choose them judiciously, easily, the one that we are interested on. So here is some other notes. So just one second. Okay. So again, some things to note that this is this is output include PCR primer. So this software or the tool can give you the PCR primer sets, so primer size to amplify the target region. TALEN offer discounts of target as an exclusionary criterion for TALEN design. It doesn't output their location. And the Mazuhen, this tool can give the users may specify restriction enzyme manufacturer for sites in the target amplicon. And the other tool, this tool can also allow primer design as well as specification of the minimum size differences of the amplicon digest product depending on the type of analysis to be done, prognosis. And then the Septa, again, this could include like a design parameter of icon. Predictive activity. Septa allows a user to specify a particular location for the cut, and you can choose a location, right, of where to cut. And this TALEN NT selection parameter is made simply by annotated pull-down menus. Then the TALEN designer also provides a genome browser for human, mouse, rat, populated with pre-designed TALEN site. So this is like a more little advanced. So it has like already some pre-designed TALEN sites are already there. Then TALEN offer is also found at this particular site, and information presented here is on TALEN. This one offers predicted offset targets for the user profile TALEN designs. This in our discussion, so we have seen like it has very great promising applications, right? So in one line, one sentence. So what we have learned is that this is basically some kind of technique by using which we can introduce double standard break at a desired location, precise the location mainly, and so that we can either go for say knockout, we can go for knock-in, or we can put some reporters in over there. We can also use it for suppose activate the gene transcription, overexpress the gene, or transcription suppress gene, and so on. So therefore, so this TALEN is like over a period of time, it has become say easy to design because now because you have also access to so many computational tool or software tools. So therefore, it is easy to design, short time to manufacture, and can be multiplexed on the order of hundreds at a time. And it have fewer constraints on the genome side, and it can be easily targeted across the genome. So it is like a because it is so moment. So if our design construct can bind to the desired location, so at least in theoretically, it is possible. It will create or it will cleave a DNA at the only at that site, not somewhere else. Of course, there are some challenges could be there. For example, centromeric region, which is like a highly heterochromatinized region. So it may be a little difficult to get access to those heavily packed region. But as such, normally we don't do any kind of modification in the like a heterochromatic regions. Like, so we all know that the eukaryotic genome, we have two parts, heterochromatic and the heterochromatin and the euchromatin. So most of the time we are focused in the euchromatic part, where the gene is like active, and then this is like a less off-target effects than the ZFN. That means non-specificity, chances of becoming or chances of mutating a non-specific target or going to somewhat else is also less, right, as compared to that. And so you can it is because by creating you can do some modification like heterodymer mutations, and also less cytotoxic for the host cell. Then it's like a comparatively safe. Knockout is easy and fairly straightforward. The high rates of the cleavage activity and essentially limitless targeting on a range of talents. So some problem could arise, like it could be also sometimes difficult to deliver. Sometimes it's like the common problem in many of the times when we do our transformation experiment. Well, we ourselves noticed that because of some reason, it is hard to get transformed. Right? That means sometimes it's difficult to deliver inside the cell. So it is like always happen with any kind of tools, and then express in vivo because of last slide. Knocking in a gene with this technology is also more sometimes could be also complicated. So this also got was recognized like methods of the year in 2011. And because it's for a NASA highlight, if you go there, so you'll find the ability to introduce that targeted tailored chains into the genomes of the several species, make it like feasible to ask more precise biological question. So it is like you can ask a range of biological questions, like what is the regulatory mechanism, what is the biological function of that particular gene, what is the like cellular role, etc., etc. So in our animal biological research method, and to scientists who develop them, we have chosen genome editing with engineered nucleases as a method of the year 2011. So it is like a one of the big success story, and that we got like a method of year in the 2011. So over a period of time, so it has been again utilized to so many expanded application to so many different organisms, including like yeast, fruit fly. So one can say the yeast or the homologous recombinations anyway easy, and without before existence of TALEN. So people used to do modification or target the yeast in a long time back, because as I said, just having a sequence assembly only 20 base pairs sufficient. We don't need anything else. But still, TALEN also give like additional boost. Right? It has tremendously helped to understand the gene function in yeast. Fruit fly, it is the Drosophila melanogaster, roundworms, cricket, zebrafish, frog, red pig, cow, etc. So people working in like a various field, they could like think of using this technique, and presumably that technique will continue to extend the additional organisms. Right? So this is like an enormous potential. So TALEN edited full-grown plants with resistance to pathogen. So for example, in the Xanthomonas example just now, sometime back we have discussed. So Xanthomonas is using this type of protein basically to escape from the plant defense system, but the same thing can be utilized against the bacteria itself. Right? So if the same technique is like utilized to make a transgenic plant or the rice resistant, so what might happen is that this plant will essentially modify or the mutate this type T secretor system itself, and then the bacteria will not able to cause any disease and so on. And so it will be like a utilized against the bacteria itself. Then full-grown TALEN edited pigs and cattle, the mutation of the growth hormone receptor in the pig cells, so-called micropigs. Then sugarcane. So some people also work in the sugarcane derived biofuel production field. So and again in 2015, these TALEN edited CAR T cells were used to eradicate tumor cell and cure to shield and etc. etc. And TALEN introduced homologous HDR. Then in human, pluripotent stem cell or somatic cell will also be achieved. TALEN have primarily been used to disrupt human genes by introduction of NSEJ that we discussed in the previous slides. So introduce indels to coding sequences. So this has wide range of applications. That we have discussed over. Now, again, among other type of applications, we can also, you can see take some example. For example, you want to do kind of tagging, okay, for the visualization. So in this example, I can see there is something you can say M-cherry. So M-cherry is like a red fluorescence. So if somebody is working with say fluorescent or confocal microscopy, and you want to suppose study the localization of the protein, okay, so whether it is localized to membrane, whether it is localized to cytosol, or whether it goes to nucleus or not, then you can label the protein. So if you want to label it M-cherry, then in the fluorescence you can see like a red fluorescence, right? M-cherry. And so you can tag it by using either M-cherry. ZFP is another popular example, green fluorescent protein. So any kind of tagging you can do by using this tool, TALE-based tool. Then we already have seen this tool can be utilized for say transcriptional activators, and so these are some examples. So several examples are there. So we have just discussed one or two, and then also this system, if you compare with the TALEN, if you compare with the other type of genome editing tools, like for example CRISPR. So CRISPR will be covered in this already there in this QIP. So if you just compare both this editing tool, so you can see that as far as the gene tagging concerned, or gene tagging type of modifications, so gene tagging means, so it is like you get fusing it to a tool. Just now we have talked about M-cherry. So similarly, for example, if somebody is interested to tag with luciferase or the ZFP for their purposes, so you can use anything like TALEN, CRISPR, right? Donor DNA is required. So gene mutagenesis, you can use both the TALEN and the CRISPR. And then similarly, for other activation as well, for other activities like gene knockout, gene activation, gene repression, safe harbor knock-in. So all those various type of application as well, you can use either TALEN or a modified version. Modified version TALEN directly, modified version TALE transcription factor, right? And that one example you have seen, or you can use this one as a for repressor and so on. So now, then if you compare with respect to the CRISPR, in case of the, as far as for say recognition, for case of TALEN, it's a protein DNA. So protein goes in there, so it and bind to the DNA of the target sequence, target DNA sequence. In case of CRISPR, it is like a RNA DNA binding on DNA. So in case of the, if the target is methylated, if the target DNA is methylated, so in case of TALEN, it will be sensitive, right? Sensitive. Methylated, just now we have seen that if suppose your target is heavily methylated, suppose heterochromatic region, then it is a little bit difficult. In case of the RNA DNA, it is not a problem at all. Okay? So RNA is not getting methylated. Then chromatin structure point of view, obviously this TALEN is sensitive. So whether you are designing TALEN to a heterochromatic part, whether designing to a eukaryotic part, so depending on the methylation status of the epigenetic chromatin structure, so it could be like a sensitive. So similarly, so is for the other one. Then off-target, less observed off-target. That means chances of getting non-specific deletion or non-specific double-strand break is somewhat low in case of TALEN. And then in this system, there is slightly more higher rate of off-target, and multiplexing is like a rarely used here and is capable. So in summary, so in last in this talk, we have discussed about one genome editing tool or a TALEN, and that is like introducing the main purpose is a genome editing and then introducing targeted double-strand break. So then once you introduce the double-strand break, the rest is like it depends on the researcher's choice, like what he or she would like to do, whether it is like a knockout cassette, one knock-in, knockout, or inserting one say fluorescent tag or something. So depending on the purpose, if that double strand break can be utilized. Right? So TALEN comprises a non-specific copper nucleus domain used to like customizable DNA binding domain, and that DNA binding domain is composed of like a highly conserved repeats derived from transcription activity like effectors, which are like a protein secreted by Xanthomonas type of bacteria to alter transcription host plant. So this origin is from the bacteria, and TALEN have generated an interest and can be rapidly designed by researchers. So now this has moved like beyond bacteria. So you have people have designed so many different types of tool, both molecular tools as well as the computational tools, and now it has been like widely applicable for so many organisms. So finally, I thank you all of you for your kind attention and giving your valuable time. And I would also like to thank Mr. Bora for giving me this opportunity for this for delivering this talk. And I would also like to thank my student Krishna for helping me in preparing the slides. Thank you so much.
Thank you so much, sir. This is Adhiraj Nath, Professor Utpal Bora, sir, student. Thank you, sir, for such a wonderful lecture. We learned a lot. It was a detailed talk covering both aspects of wet lab and dry lab in very detail. So now I would like to ask the participants if they have any questions for Professor. So as I can see, there are.
Yeah, so yeah, we can just discuss it about, as I said, I'm not at all expert in this field, but we can discuss together. Yes, well, please start. Yeah.
So there is one question for you, I think. So how are talents delivered into plant cells?
Okay.
Okay.
Yeah, so for gene delivery system, so this basic gene delivery system is same. For example, people those who are working with, for example, we work in neurospora casa. So whatever technique we follow, it is electroporation. So mammalian cell is transformation. Similarly, for plant cell, it is agrobacterium based, like I said. So it is you can have like a nice. So when I remember when I was working with plant cell, we used to cut like tomato leaves into small pieces and then make the construct with agrobacterium derived based vector, and that was used to deliver the DNA. Okay, so maybe some people have used some other technique. I'm not familiar, but I myself used one long time back. This agrobacterium based plasmid system. Yeah.
So thank you, sir. May I ask if there is any more question from the participants? Okay, I don't think there are just appreciations for you. Everyone is saying thank you for this lecture. Of course, it was a very wonderful lecture. We learned a lot. So thank you, sir.
Thank you so much. Thank you all.
Okay, sir.
Have a nice day.
So,
yeah, thank you.
Okay, sir. Thank you, sir. So with this, we come to the end of our lecture, I think. So for tomorrow, there will be a new link for you. And there will be a practical session tomorrow, which will cover some aspects of designing targets in DNA, which are novel, suppose which has not been shown before, which doesn't have any database or any tool. So how to approach for that? So that will be a short practical session, and I'll be handling it. So if you want to follow along, you can install Python, Flask in your laptops, along with NumPy and Pandas. So I'll give the details over the group, and we'll start at 12 tomorrow for this practical session. And it will be a part of that homing endonuclease talk. So thank you all. Thank you for joining, and see you tomorrow then.
Thank you, sir. Thank you.
Okay, sir. Thank you, sir.